This is a Founding Media podcast, produced at Austin Community College District.
Welcome back to Science of the Mall, y'all. I'm your co-host, Dan Dillard. Alongside my co-host and the director of Austin Community College's Bioscience Incubator, Nancy Lyon. Today, we invite Jolie back onto the show, and she's joined by her boss, Josue Moran, who is the co-founder as well as the VP of Research and Development at Angstrum Bio. Angstrum Bio is developing a more efficient, more sensitive viral testing method, and Jolie and Josue explain exactly how they are accomplishing this important feat. They talk about what it was like to start a biotech company in the early days of the pandemic, and how Austin bioscience community and entrepreneurial spirit helped them to build their product and grow their business. They share some valuable insight into the importance of networking and proving yourself through internships, as well as getting yourself plugged into your local community by taking advantage of resources like those found at ABI. It's a great episode full of insight, so let's jump right in and learn about what Angstrum Bio is doing here in Austin.
Let me give you a brief background on Angstrum. So we founded Angstrum in November 2019, originally as an antibody engineering shop, essentially designing antibody drug targets on the computer before you actually make them in a lab and try to test them into clinical trials. And so as we started that company, we entered into 2020, and like many other companies had to do at that time, we had to do a pandemic pivot, as they called it. So we transitioned into in vitro diagnostics, obviously using COVID-19 as a beachhead into that front. So we made the pivot, and we sort of tried to find a lab that would take us in and allow us just a small amount of lab bench to initially.
Wait,
test
that's what I do for a living.
That's right, but we couldn't afford it at the time. So we started this. We were able to through some connections here in the Austin community that we're really grateful for. We were able to find a small lab bench inside of another local Austin biotech company called Tevito Biosciences. That's how we started Angstrum, and that's how I first met Jolie, who was at that point part of Tevito Biodevices.
Very cool. So I'm gonna back us up. I know you mentioned it in this explanation, but I want to touch again on what Angstrum does, but also explain it to us like if we're freshman biology students.
Like high school freshman biology students.
High school freshmen. Okay. The platform technology that we developed is really based on three main principles. The ability to barcode DNA, use DNA barcodes to tag or flag each individual patient sample, then run that through what's called a DNA sequencer. So you're able to detect every single nucleotide of any potential virus infection within a patient sample. And then on the back end, which is what a lot of my co-founders have worked on, are the algorithms to deconvolute some of that data, right? So what we're able to do, and the real value proposition here, is that we're able to batch potentially tens of thousands of patients into one small little test tube this big, and run them through what's called our Oxford nanopore sequencer. And so we're able to detect every single nucleotide, not only of a potential viral pathogen, but also of what we call the DNA barcodes on the ends of what are called the primers, on the ends of every primer for every single patient. And we're able to deconvolute that on the back end, and do that at a very, very large scale. One of the things we noticed early on in the pandemic was that a lot of the testing was just way too slow and lacked a lot of sensitivity. Mm-hmm. And this is a platform technology that can really, really deliver scale and sensitivity.
Can you, I know what you're talking about, but barely. So I'm going to say you went to like senior college. So like, but no, seriously, like just because I think the people that are listening, like they don't understand, like put it in the context of COVID. I mean, it's a great example, but like, how does the normal test do it versus yours in like just like, take the science out of it, whatever, right?
Uh-huh. So the normal test, so part of the foundations of the normal test, say the PCR test, right? It detects viral RNA inside of a potentially infected patient, amplifies that many folds over different cycles,
makes lots of
and
copies.
makes many copies over many cycles. And there's a color, like what's called a little flag. We'll call it a color that increases over time, and it creeps up. And that color change and increase in color change over time allows you to detect a potential positive patient sample, right? And so that's one at a time, and you're limited based on different colors that you can really do at one time, and you can actually visualize at one time. What we're able to do is eliminate the whole color-based reading, and we're able to group many thousands of patients at one time. And not only the value proposition is not only that we can pool so many at one time, but the readout and the information that you garner at the end is so valuable because it can also potentially detect viral variants. It can discriminate between flu A or B and say RSV. So because you're able to get the genetic sequence at the end. And that's where the greatest value proposition lies. Imagine being able to do many, many tests for one patient at one time in a single well, as opposed to having to separate and have to do many samples for a single patient. That's where the value comes in of our test.
So then, like, if you did a bunch of people, let's say like a high school, and you put everybody's in there, and then this DNA barcode that you're talking about, that's how, like, it's got like Nancy Lyon on one of them, and so if that's the one that comes back positive, you know who in the school is positive, or?
That's exactly right, Nancy. And we assign you a DNA barcode before you even go onto your sample.
GGTAAC is my favorite one.
We'll design one just for you, Nancy. So everyone gets a unique barcode assigned to them before the sample gets processed, and then we're able to read that out on the sequencer using our proprietary algorithm.
Cool.
And that's the value. And the other thing is that, we can deliver a test within 24 hours. So the time component to deliver a high throughput test with a lot more information than standard PCR at an affordable cost and a faster turnaround time. That's what we need. That's what the pandemic needs. That's our vision for in vitro diagnostics and where we think that needs to go.
Right.
Well,
that'd have been nice.
Sounds like it's going wide and deep is what I'm hearing versus, spreading yourself thin.
That's exactly that.
Getting all that information.
That's right, Dan.
Very cool.
Absolutely.
So you said you founded Angstrum in 2019, prior to the pandemic, and then you had to pivot in a big way to the pandemic. Can you talk a little bit about what happened during that turning point, and also how ABI played a role in that transition?
Absolutely. So that transition was pretty rough. We were, as any startup knows, you're trying to raise some money as a software company, and then you realize that that's not going to be the direction to go once this new pathogen really hit the world at just a rapid speed. So we founded Angstrum, like I mentioned. I have 4 other co-founders: Carlos Santos, Jonathan Feldman, David States, and Forrest Cox. And, we kind of put our heads together. Carlos had an idea of something that he was kind of brewing, of a way to do massively parallel diagnostics. And that's actually where our name comes from, AMP. So Angstrum Massively Parallel Diagnostics, right? And we had this idea in our head that we could pivot. We could either continue down this one path, or we could pivot using as long as we could do a proof of concept somewhere in some space that would take us in. And what's funny is that I think what's really humbling at that time is that all of these ideas are sort of up here, right? They're very theoretical, and will this work? Can it work? How can we de-risk this as much as possible? And part of that conversation, part of that decision, actually was driven through conversations with Nancy, for example. Nancy, who do you know? Do you know anyone who has just a small shot? And ABI was actually closed at this time. They weren't even allowing anyone to go in. So do you have anyone? And you have to really take context here, because do you know anybody within the context of this global pandemic, any lab that would let me come into their space and do an initial proof of concept work? I need about 2 feet of space. I just need to test something. I'm a
little guy. I take up much space.
And it'll only take, the pitch there is, I only need a month. I only need maybe 2 to 3 weeks. And here we are 2 years later, and we're still.
It's always the case.
And I think ABI really, and really to Nancy's credit, really played a key role in garnering those connections, right? And that's something that we hold dear. And that's actually one of the reasons my other co-founders who started another company here in Austin, one of the reasons we moved the entire lab with that other startup to Austin was because of the connections, because of the relationships that we saw here in Austin, the Austin ecosystem that Nancy was a part of and is a part of. And we really saw value in that. And I tell you what, during the pandemic, we really saw the fruits of that. So it really, really speaks to Nancy and how interconnected she is and how well connected she is and how she's able to just find a way for people. And I think I may not have ever said that to you directly, Nancy, but we're always very, very grateful for that and for you.
The story that reminds me of is, so these guys came to town. It was like Dell Medical had a lab space. We had it, Temple, all of these places. And these guys were coming in from out of town. They're like, oh, there's this company from St. Louis that wants to move to Texas and they want to take a tour. Okay, so we invited them over. And they had already set up an appointment to go see the school, the med school's labs, ours labs. I think they'd had a phone call with Steve Frazier down at Star Park. I'm not 100% sure of that, but there was this conversation. And when they came over, there's a networking event twice a year. BioAustin has a BioBash. It's actually coming up. It's like in a couple of weeks. So if you haven't gone to BioBash, go to BioBash. It's a great networking event. They only do them twice a year, so they're really well attended, really fun. And it happened to be that night. And we told these guys, like, hey, you should go to BioBash. It's really fun. And they did. And by the time they ended up, they're like, oh my God, we met so many cool people here. We're telling our funders who want us to move to Houston, I think we're going to move to Austin. And they did.
That's exactly right. That was
everybody.
No, and we all saw the like we said, the ecosystem, the tight ecosystem that was here, no
was
no
that
regrets.
What drew you to Austin's bioscience community?
Absolutely. And I think my other co-founders would echo the same.
They were dreading the idea of going to Houston. They're like, but, and Houston's got a scene. I mean, it's got its thing. Some people would say they've got more of a life sciences thing than we do, but they got our thing.
So, talk a little bit more about the cooperation and collaboration happening in the biosciences community that you've experienced here. I mean, you said this networking event, but since you've moved here, what else have you experienced?
Wow. So I think the most telltale, I guess, collaboration within for me within the Austin ecosystem for biosciences is really this collaboration that we have with Tevito. And I don't know if you want to continue expounding on that, but I can really talk at length about that relationship. Sure. That's one of them. I think the fact that Laura Bosworth, she's the CEO of Tevito Biodevices, really allowed a group of scientists that she's never met before into her kindred lab space, while she's also trying to keep her own biotech company afloat and running, and also saying, sure, I can be this Airbnb of life sciences for you, and I'll show you that I'm a super host as well on top of that. And because we have, like I said, we started with this really small footprint there. We said we'd only be there a month. That kept going. The relationship there was I think a win-win, because she was also launching COVID testing at the time using a different platform. There were times when we would work together to troubleshoot execution and launch of that platform, of that technology, of that COVID testing. It was sort of a standalone COVID test. We really worked together to figure out problems really, really well under the context of this global pandemic, without butting heads. It wasn't competition. It was just really good science, really good business. And gosh, I hope that's just a bellwether of things to come for the future of Austin life science ecosystem.
I think it's a testament to all of you, all three of you, Laura, Jolie, everybody. I mean, when this happened and everybody was freaking out, going, "Oh, I want to help," Laura actually put it out there, like, my lab, I will move out of my lab if somebody that's working on COVID can find a good use for it.
Wow.
And nobody took her up on it. Like, I mean, Angstrum finally did, but nobody took her up on it. She literally put it out there like, I won't even charge you rent. I mean, obviously, I'd like to get my rent back, so I'm not burying myself further in the hole. But for the cause, if somebody can jump in here and save the day during COVID, my lab is yours.
And to add a little color to that, you're right. She didn't charge us rent. She knew that we had zero funds. We didn't have anything for months. Nancy let us borrow some pipettes. We didn't even have pipettes. The way that we came together during that time is really gets a lot of us emotional. Just the way that came together. Yep, it really does.
Jolie's sitting there silently just listening, but she was had her fingers in every ounce of that.
Yeah, I wanted to ask Jolie a little bit about the experience of coming on board.
Well, we had already worked together for a year in that we were in the same space, and Josue really helped us, gave us advice if we had problems or unusual results. He had ideas as to how to help us with it. We shared materials, different little viral particles.
We shared viral particles.
We did. Well, they were synthetic. They weren't harmful. But where to find materials because things were scarce back then. We had really sort of worked together already for a good year, and I could see what they were doing was just the neatest thing. They were doing sequencing on such in this tiny little instrument. I know Nancy at ABI, the next gen sequencing instrument is pretty big, the Illumina. But at Angstrum, they just do everything on this tiny little nanopore sequencer that's about the size of a phone. So just the product is really cool, and I was very interested in what's happening in the invisible world there, and what's happening with the DNA, what's happening with the enzymes and the RNA, and
so it was just the passion of learning new things. It was, at the end of the day, it was just something.
And being excited about PCR, which just cracks me up because I'm like, oh God.
Yeah.
Yeah. I think it's worth mentioning that, Jolie did say that, we had worked together for a year, essentially in the trenches together, right? Sharing
COVID trenches, really.
Yeah.
It really is the COVID trenches, because, the others will tell a similar story, I'm sure. Like, if you speak to people who have been working on COVID, people haven't slept, right? They have not in the life science space. Many times they're 2 o'clock, 3 o'clock, 4 o'clock in the morning, getting something to work. I'll come back. I usually do an earlier shift, 5:30. I know she just left just because I could tell that somebody had cleaned a biosafety cabinet and put their initials there at 4:30 in the morning. Things like this that really don't really get told very often. And you see people work. You work alongside people like this, and you admire the breadth of knowledge, and you admire the grit. And I tell you what, you can't teach grit, right?
It's totally true.
You can't teach that, and I think we've all seen that in Jolie, her fund of knowledge that came from ABI, the grit that she has, and we saw it as a real win-win for a transition over into Angstrum. Of course, Laura certainly values Jolie and holds her up to a high regard as well. we are passionate about career development. Laura knew that. She knows that I'm passionate about that. And like I said, we saw it as a win-win in that, one, it put Jolie on a career trajectory towards next-gen sequencing, which she was really passionate about. Angstrum was still in the Tavito space. We're still there now. The tribal knowledge that Jolie has in Tavito also could stay there because we were still in the same space. So, and that has really, I think we've seen many examples of that, Jolie, where you have, certainly Tavito has been able to lean on you as well based on your experience with them, and because you're still there.
Yeah, sounds like
and it works. It is, and it works really well. And that goes for the entire Tavito team as well.
Yeah, I think for as far as the hiring process here, sounds like, and you correct me if I'm wrong, Josue, but being able to work for a long time with the person that you hire, you as you mentioned, you've already kind of experienced that working conditions and who the person was, the grit, all of that before bringing them on. But going forward, you guys have hired other people, I'm sure. Is that correct?
That is correct. Okay. I think we've hired one other individual, Piao Yi, who is a lab tech 4 in our lab. One of the things as a startup is, I won't take credit for this phrase, but Mike Makebulls, who is a VC out of called Floodgate out of California, has a show, and he starts his show and he says something to the effect of, being in a startup or launching a startup requires bringing out your inner Wonder Woman, your inner James Bond, your inner MacGyver, right? And as a founder, as a co-founder, that is absolutely true. And your next hires after that are just crucially as important, because you want to try to find people who also have that inner Wonder Woman, that inner MacGyver. And for those of you who don't know what MacGyver is, you have to YouTube this and watch the show. But yeah, it's about figuring things out, unlearned, never trained in something, figuring something out on your own with the very little bits that you have, right? And saying, how do we put this together and survive, right? And that's why your initial hires are super important. And you wonder, how do you find people that way? Jolie was certainly one of those individuals, and it was very clear from the outset of my experience with her. So we value that inner Wonder Woman and MacGyver in her.
I love that analogy, because a lot of times it's like, you've got to wear multiple hats, but that's just like overused all the time. But the MacGyver analogy, having to make do with what you have, super important, especially getting started.
It is, and that speaks to my other co-founders as well. I have to give all of them a tremendous amount of credit.
So for students listening, I'm just curious, what maybe give them some insight on how to best prepare to be that inner MacGyver. What would your advice be?
Can you train that?
And this question is for both of you.
Do you want to go first, Jolie?
Well, I think for the students, when in class, there are always a number of people trying to do one essay, one test. So I would suggest that you be the one to, immediately volunteer to try to do it. Get your hands in there and learn how to do everything. If there is a small company that you could intern or volunteer at, just so you can start getting experience. I think smaller companies are ideal because, again, you do have to wear a lot of hats. I know it's an overused phrase, but it's true. That way you learn a little bit of everything, so you can pull all the information together as to how to solve one problem.
But do you think that that's personality-based? Like, do you think that that's something you can teach, or do you think that's sort of just like, that's who you are?
I think you can train yourself to become that kind of person. I think that if you just keep trying and put yourself out there and don't worry about what anybody thinks, just keep putting yourself in those situations. You can become that if that's what you want.
There's a word that comes up for me based on what you said, Wonder Woman, MacGyver, and what you just said, Jolie, and that word is fearless. Sounds like you've got to be able to work on those fears to fail and whatnot, just to go try new things. And so once you've overcome that fear, you have that grit to to do things that are valuable for especially entrepreneurial employees.
And I do want to say it's also worth it. I get to work with people that are just so intelligent and conscientious, and I don't know if it's the particular. I've been so lucky working with Nancy at ABI and Laura at TVDO and everybody at Angstrum, but there's just a particular joy in working with people that you respect, and it's just been the best field. I'm so grateful to come into biotechnology.
You know, to answer the first part, is it trainable? Is it coachable? It can be with good mentorship, and I tell you what, you have to. One of the biggest problems, I think, at least in education, is that students aren't allowed to fail often enough, and they're not permitted to make a mistake. And on a layer underneath that is that when we learn things very linearly. Somebody had this problem and then they found the solution, but you don't know what happens. You know, there's actually a good TED talk from Ari Eisen out of Israel. He talks about being in the cloud, going from A to B, and you don't understand all of the ups and downs and the turns that happen to get there. And when you learn science, when you learn how things were discovered, you don't learn about all of the troubleshooting that happened to get there. And there aren't enough practical, I think, experiences that allow students to fail leading up into high school, into college. So when you get to graduate school, for example, in a graduate program, you're often required to figure things out on your own. And there's a lot of, and I'll say this, and I don't mean this is actually probably for another talk altogether, but there's a lot of depression that happens within graduate school because of the fact that you have never failed so many times in your life, and you have nobody to hold your hand and guide you, and you have to figure it out on your own. And if you're not told it's okay to make a mistake, this is part of the process, right? This is the way. Then you kind of say, okay, now I can have some self-compassion and say, this is fine. We can continue on in this route. I can continue failing as long as I change some variable along the way to potentially give me a different outcome. So you can teach it. It's just, it requires good mentorship. You can teach it. It just requires good mentorship.
I think the word to replace failure should be testing.
Yeah,
we're gonna test this process to see if it works, then move on to the next test and just kind of continue. But yeah, I think language. I think you're right on to something like language and the fact that during your first 12 years, you've got that one answer and that's it, and you move forward and you're graded on this pass fail thing. If you fail, it's considered bad versus learning. So
right, and you learn so much. You learn there's. And I've said this many times. I know probably Jolie gets annoyed, but there's a few ways to know the truth. To learn the truth, you can either be told the truth or you can experience it, right? Mm-hmm. Experiencing that failure will help you learn the truth way more than just me telling you. So there's value in somebody making a mistake in an assay. And don't get upset because you have that. There's so much value in that mistake learned that you said, man, I just messed up. And this has happened to me, man. I've messed up 2 or 3 days worth of work.
Yeah.
But man, tell you what, I will do everything I can to not make that mistake again.
I think people also don't realize, like, in research and development, like R&D, like grad school, right? These PhDs or whatever, in order for them to get this, and what they're doing is doing something that's never been done before. You might use techniques that other people have used to get at this, but they have this unique problem and they have to figure out how to answer the questions. And it's often like you're the first person that's ever even thought of this, and you're going to screw it up a lot.
Mm-hmm. That's right. And there's plenty of parallels to startup.
Yep.
Right.
They say that about Austin, like, there's an entrepreneurial mindset. And I think it's what Jolie was describing of just like, you just got to get in there and try and
roll your sleeves up.
Yeah. I think there is sort of a personality type and probably, like you said, a mentored person that can take that.
And the whole community has a lot of people in that same mindset. And so it makes it okay because people, when they're having coffee or having conversations, they understand each other. It's not like maybe other cities that are less entrepreneurial. So that gives more permission in the cities, this great city.
Right.
Any advice for folks that are listening who are considering a career in biosciences, like what they should be thinking about or what they should be doing, or just any advice at all?
I tell you what, the first step, I think maybe we can bucket this two different ways. The first is for people who are interested in finding a career in biotech. And the second one is once you're in it. So first, networking. I know people say this so often, but you really have to get out there and get to know people and meet people. Ask for coffee chats. You have to understand that people really like talking about what they do. If you can just put yourself out there and say, hey, could I, bother you? Could I do a 15-minute coffee chat with you, a Zoom coffee chat even, just to get to know how you got here, a little bit more about you? People will carve out 15 to 30 minutes of their day to have a conversation with you. Find networking events. Get out there and network. Show that you care. Show that you are willing to learn, right? The second thing is once you're there, be malleable. Put yourself out there. I think Jolie mentioned this point earlier. Volunteer to do the extra work, to learn, because it's only for your own edification, right? Mm-hmm. Volunteer to do that. It's okay to make the mistakes, but put yourself out there. Have a voice. We live in a world where the squeaky wheel gets the grease, right? Get out there and talk to folks. One thing I recommend to anyone going into the life sciences as well, understand and learn some kind of data management methods. There's simple ways to do that online via Coursera and other e-education software, something to learn how to organize, manipulate data, or maybe some programming. Very valuable. I think the scientist of the future is not just the one that can just do a lab assay. Scientist of the future is someone who can wrangle data, find the details in the data in your own analysis. Super, super critical, right?
What about, I'm curious as to that from the company standpoint, like obviously we're at ACC, so community college, and the people that we are interacting with and the students that are going through here have 2-year degrees. And I know that I've been to meetings with other people who have said, the hard part is to get these employers to realize that a 4-year degree isn't necessary for every single job. And there's definitely some bias in education toward like these graduate degrees and all these degrees. And is it necessary? And how do you feel about that from the perspective of an employer of like?
You know what, you've seen that. I've seen that there's certainly value that comes out of programs like ACC. I think what happens, because there is that certain stigma, I think there's an added layer of work for somebody to say something along the lines of, let me do a free internship to see how I can perform, see how I do, see how things happen. And that's actually what I did. I volunteered. I did an internship, a non-paid internship in a biotech company in 2009. And I said, while I was working full time at a consulting firm, and I said, give me just 1 hour of your day and I can come over and I can help you with anything you want. So I took my lunch break from my current company, from my consulting company, drove 5 miles down the other side of town, took an hour and a half lunch, and I worked for the other company, for the biotech company, as a way to say, hey, I can do this work. Just let me show you. You have to prove yourself, whatever it takes. If you really are passionate about getting into the space, you have to do these kinds of things. You had to do it back then in 2009, probably still have to do it now.
For the students that are introverts, like myself, going to a big social networking event is a little scary. So I think one way to start is interact with your different teachers, even if teachers that are not your own teachers, and start asking them, can you explain how to do this? Or I knew, Nancy, you told me about Dr. Kisao, and I knew that he was an adjunct professor who was not one of my teachers, but I knew that he was hosting He was going to be at the lab to do a study group kind of thing. So when I wanted to learn about PCR, I just brought him all my questions, and he was eager to help. So I think really interacting with your teachers and the people that maintain the labs. I got my job at ABI through one of the teachers and you, and I think that they know people in the industry. A lot of ACC teachers are working in the industry, so they know where the jobs are and who you can be connected with and where you might fit.
And, and to Josue's point about the coffee dates, like I had a woman reach out to me on LinkedIn the other day, and she's a postdoc over at UT, and she's realizing that maybe it's time to try to make a transition in the industry. And so she said, I just want to meet you and hear how you made a transition out of UT. And I, linked into her and said, hello or whatever. And then I kind of accidentally blew off that part of it. And she chimed in about a week later. She said, I hope you don't mind. I'm going to bring this up again because I really would like to just schedule 30 minutes or so. And we had a great conversation. And when she started telling me about the work she's doing, it reminded me of this company that has recently pulled out my application, who is going to open up here. And so I sent him a note. I said, hey, are you planning on hiring somebody when you get here? And he said, yeah, I think so. Why? And I introduced the two of them, and I don't know what's going to happen, but it, here's this woman who literally cold called me and kind of had to nudge me to get me to do it. But it may have landed her a job.
Right. Well, you just never know. 100%. Oh, that's an awesome story, Nancy, for sure.
And it's shocking how often it happens. It's just like everybody needs something. They don't even know what they need. But the more people the more conversations you have.
Well, Jolie and Josue, it's been a wonderful episode. We learned a lot about what you guys are doing, but also processes of getting into this space. And so it's been very valuable. Thank you very much for your time.
Thank you, Dan.
Absolutely, Dan. Happy to be a part of this program, this podcast, and with thank you to you too, Nancy.
Sure.
Thank you so much for listening, and thank you, Jolie and Josue, for joining us on Science in the Mall, y'all. It was inspiring to learn about Angstrum Bio's journey and the way our community here in Austin came together to collaborate, cooperate, and fight COVID. And it's always so helpful to hear advice about how to break into an industry. From networking to internships, there are so many great resources in this great city for folks looking to work in the biosciences. And if you know someone who might benefit from the wisdom shared in this episode, share the wealth and send it to them. Thanks again for tuning in, and join us on the next episode to hear more about the intersection of entrepreneurialism and science as we learn about another startup that planted roots at ABI and has gone on to find great success. This has been a Founding Media podcast, produced in partnership with Austin Community College's Bioscience Incubator, which is the region's only wet lab space for Austin's growing bioscience industry.